All right, we're going to discuss next the actual activation of T cells. All right, and the process is going to be a little bit different between cytotoxic T cells and T helper cells. I'm going to start with the T helper cell. All right, and in terms of our T helper cell, all right, what's going to happen in order for that naive T cell to be activated? It's going to have to encounter an antigen-presenting cell. And remember, those antigen-presenting cells are going to be cells like macrophages, dendritic cells, possibly a B cell, although in the terms of T helper cell, most often it's a dendritic cell or a macrophage that does the activation. All right? Remember that phagocytotic cell engulfs those particles, chews them up, digests them, displays our antigen, which is the little orange dot here, out on the surface of the cell. It wanders around and eventually that antigen-presenting cell runs into a naive T helper cell. That interaction then is going to happen between major histone compatibility complex 2, the T cell receptor, and the CD4 on the outside of our T helper cell. All right. Once all of those locks into place, what's going to happen is you're going to have that T helper cell secrete the cytokines interleukin 2. All right. And that interleukin 2 then is going to be what activates our T helper cell. All right. But in terms of that, the interaction between the antigen-presenting cell and the T cell has to happen in order for the interleukin, the cytokine, to be released. All right. Then what happens with our T helper cell? It will differentiate into either the Th1 or the Th2. All right. And then it'll go off and either now activate B cells or stimulate proliferation of our cells like our macrophages and our phagocytic cells. All right. So we've got a complex there at the cell membrane that has to come together to release the cytokines. And then that T helper cell is going to go off and activate parts of the immune system like the B cell or increase phagocytosis with the phagocytic cells. All right. Our cytotoxic T cell is going to be a little bit different in its interaction. All right. The cytotoxic T cell is going to essentially involve a complex between CD8 and the major histone compatibility complex 1. All right. That's one of the first differences. Okay. In terms of that, typically what we're talking with cytotoxic T cells. Okay. Cytotoxic T cells are going to target typically eukaryotic cells. All right. These are going to be eukaryotic cells infected with virus, or this can be eukaryotic cells that have become cancerous or are growing out of control. Because what happens, if you have virus actively replicating inside this cell, you've essentially got the antigens inside the cell. What that's going to do is you can kind of think of it as modifying our MHC1 a little bit. And in terms of the cytotoxic T cell, all right, it's going to be looking for odd MHC1s on the outside of cells. when it encounters that odd MHC1, because essentially the way that MHC1 becomes mutated is because of the infection or the antigens that are inside the cell. All right? CD8 will come along. What that's going to do then, that's going to activate our cytotoxic T cell. And how cytotoxic T cells are different from the T helper cells is inside those cytotoxic T cells are molecules known as perforins and granzymes. All right? These are essentially enzymes that are going to kill our cell. All right? That cytotoxic T cell is going to release those perforins and granzymes. Those perforin and granzymes are going to be released into our infected cell. And what's going to happen? Okay? Those perforins and those granzymes are going to trigger cell death. All right? And in terms of the process we're talking about here with the cytotoxic T cells, let me go with a different color here. All right? The cell death that we're talking about with the cytotoxic T cells, how they kill, is by a process known as apoptosis. All right? Apoptosis is programmed cell death. Essentially, what every cell has is it has a mechanism inside of it that's like a self-destruct button. That's what apoptosis is. And what those perforins and granzymes do when they're inserted into that eukaryotic cell that's either infected with virus or it's growing out of control, it triggers that cell's auto-destruct button. And that auto-destruct button is apoptosis. Okay? So in terms of that, it's considered programmed cell death, meaning it's just that eukaryotic cell that dies. It doesn't take out the cells next to it. So cytotoxic T cells, this is what I mean with cytotoxic T cells really being more like a sniper. All right? They're targeting something specific. Once they've targeted that first abnormal cell, those cytotoxic T cells have become activated, and they're going to go off and scan for other cells that have that abnormal major histone compatibility complex 1. All right? But in terms of that, that's kind of the differences I want you to take away between cytotoxic T cells and T helper cells and their activation and their function. All right? If you have any questions, please let